Thank you, everybody, for joining us tonight for this Illinois Crayfish Finders training workshop. I'm going to start with an introduction to this project and talk a little bit about iNaturalist, and then Dusty is going to take over and do all of the crayfish talking. So this is a special research project that is brought to you in partnership between Illinois RiverWatch and Illinois Natural History Survey. And I am the volunteer coordinator for Illinois RiverWatch, which Illinois RiverWatch, if you're not familiar, is a statewide community science project that helps monitor the streams in Illinois. So this program is part of what is called RiverWatch Participatory Research Opportunities, or RiverWatch PRO. And these programs that fall under this RiverWatch PRO subtitle are community science projects that are designed to connect volunteers with researchers. These RiverWatch PRO projects are community science projects that are designed to connect volunteers with researchers or community partners to answer specific questions. So Illinois Crayfish Finders is one of those special programs. So we're running this program for 2 years, from January 2025 to December of 2026. And while we're interested in learning more about native crayfish, the primary goal of the project is to collect data on the range of 2 invasive species in Illinois, which Dusty will talk more on later. So I know a lot of you here are probably already familiar with iNaturalist, but if you're not, it's an online platform for sharing biodiversity information. And so the basic idea of iNaturalist is a user will share an image or sometimes an audio of an organism that they found with details on when and where they find it. After the observation is posted, other users can give their input on identification. And after 2 people have confirmed the identification, the observation is considered research grade. And I'm going to go through a quick run through on how to use the program for our project. And you can use the iNaturalist website or the mobile app. But the first thing you need to do is to make an account. So I have this is a picture of the website, but it's pretty easy to walk through on the app as well to find the things I'm going to be talking about. So for this one, the sign up is up here. And then once you have your account, you can start making observations for our project. To find our project, community button, and then it'll have a drop-down menu for projects, or if you're on the app, it's just on the left sidebar menu, and you'll find a thing that says projects, and you can search for Illinois crayfish.
Uh-oh. I'll go ahead and start. Let me share my screen real quick. All right. So yeah, my name's Dusty Swedberg. I'm a aquatic ecologist in asticology, which is crayfish research at the University of Illinois, Champaign-Urbana. Champaign-Urbana. I work for the Prairie Research Institute as part of the Illinois Natural History Survey. And my main focus is crayfish. I do some fish stuff too, but what I'm really passionate about and what I really enjoy doing is crayfish work. I've been doing it for about 15 years now. If some of you may have been on back, I think in October, when I gave a similar talk for the fall series, I'll kind of go over some of the same information for those that weren't here or that weren't able to attend. And then I'll kind of get into more of the specifics for this project. So what I'll focus on are kind of the key characteristics to look at to distinguish certain or different species within Illinois. I'll focus mainly on our most common species. And then I'll also show you when you're in the field or when you're out sampling or when you come across a crayfish, how the best way to handle it, get the photos we need. There's about 4 or 5 different types of photos that we need. And when I get to the photograph part of this talk, all those photos were taken on my cell phone. They're of crayfish here in our collection. So I should have mentioned I'm also the acting curator of the crustacean collection here at the University of Illinois as well. But those photos were all taken with a cell phone. So it's the great thing about the advancement of phones now is you can get really high quality images. Um, and they're good enough so that we can get successful IDs on a lot of these species. And again, it's only 4 or 5 photos. It usually will only take about a minute or two, and I can go over that. I'll go over that in a little more detail when we get there. So first, I want to talk a little bit about what a crayfish is and what we have here in Illinois. So in Illinois, there's this old Calvin and Hobbes, that's everywhere, I guess. It's old Calvin and Hobbes comic strip. I used to read Calvin and Hobbes when I was supposed to be sleeping. I don't ever remember this one, but to a lot of people don't know what a crayfish, crawdad, mudbug, yabby, um, whatever you call them, what they are. So, uh, crayfish are a aquatic or semi-aquatic arthropod that respires with gills, has 5 pairs of non-feeding appendages, which puts it in the order of Decapoda. It's dorso-ventrally flattened, so flattened abdomen, and it has direct development. So that's the scientific definition of a crayfish. In Illinois, we have approximately 25 species. This is kind of changed here and there with some splitting of species. We have, at the time I wrote this talk, we had 4 state endangered, but now we have 5 state endangered or threatened species. Those are Faxonius kentuckiensis, Faxonius indianensis, Faxonius um, placidus, Faxonius lancifer, and Faxonius, the most recent one that was just added to the state endangered list uh, either a week or two ago, is Faxonius standardi, which is one of the few um, purely state endemic species, meaning it occurs nowhere else but in the state of Illinois. And that species occurs in the Little Wabash River in southern Illinois. Of those 5, there's 2 more that are considered species of greatest conservation need, but they don't fall in the state endangered or threatened. That is Illinoisensis, which is the other kind of wide-ranging state endemic species, and Cambarus tenebrosus, which is on the SGCN list, but is not state endangered or threatened. We have 2 invasive species of primary concern, which I will talk about in detail, and it's kind of the big thing that I want to get from. Let me see what this chat is real quick. Okay. One of the big things that I hope to get from um, people going out, taking photos of crayfish, documenting them. And I'll go into a little more detail of that as well coming up. We also have 7 primary burrowing species. If you went out this past weekend after the storms or after the rain, you might have seen them out crawling around, moving around. They often come up after rain events, especially in the spring. Um, so if you went out, I noticed we had some on the iNaturalist page already on the group from burrowers from this past weekend after the storm. So that was pretty interesting. Come on. All right. Weird. As I mentioned, Illinois has 25 total species. Um, so the big things here are the species on the left. This is a burrowing species. You can kind of tell because of the shape of its chelae. It's kind of got this flattened edge that's meant for moving dirt, pushing it around, pulling it in and out of their edge, kind of manicuring their burrow. Burrowing crayfish are very particular on keeping their burrow clean and well groomed. It's their home, so they want it to look nice, is what I always believe. The species here in the center is one of the invasive species. Well, it's invasive to the Chicago area, to the northern 9/10 of the state. It is native in the extreme southern parts of Illinois. down in Alexandria County and those far southern reaches, kind of down the lowlands, and the floodplains of the Mississippi and Ohio rivers. The species on the right is one of the more widespread species throughout the state. That's Vexonius virilis. It is often confused and can be confused with rusticus because it is um, the most similar looking. But I'll go into a little more detail on how we can kind of distinguish those here in a second. So within Illinois, I mentioned burrowing crayfish. These are the ones that a lot of people have familiarity with, though they may not see them actively. Uh, you'll see these chimneys, these burrows where they've excavated dirt. These species primarily occur in anywhere where there's a slightly elevated water table or perched water table, shallow water table. You'll find their mounds, their dirt, and they spend 95 to 99% of their life in their burrow or on the edge of their burrow. In Illinois, we have 7 species. There's still kind of some um, debate going on with these. It's um, still some debate on how many there are. Um, there's been some splitting within a group that used to be the devil crayfish, Cambarus diogenes and Lacuna Cambarus diogenes. It has since been revised. Taxonomic revisions have been done, and now that species is confined completely to the East Coast because that's where it was described from. And then Illinois now is represented by at least three three different species. Nebraskensis up in the top right, um, Lacuna Cambarus chimera that is over here on middle left, and then Lacuna Cambarus erythrodictylus, which is here in the middle right. The other species here pictured are Lacuna Cambarus polychromatus, Creserinus fodiens, and Procambarus gracilis. I'll show some photos of these guys later, though these will be the only ones in color because the other ones are from museum collections. Uh, so the reason I included this slide was in Illinois, some of the streams we go to do get pretty low. So you do have the possibility of finding non-burrowing crayfish or tertiary burrowers within the streams. This is more common in southern Illinois or the Ozarks of Missouri. It's been well documented there. Sometimes when water levels, sometimes when water levels get low, uh, almost all crayfish have the ability to at least burrow down and get into some hyporheic flow. So when you see a dry stream, it doesn't always mean that there's no crayfish there. If there's some hyporheic flow or interstitial flow, they can go down to get to that water level. Their gills just need to be moist and they can breathe just fine. So when we talk about stream crayfish, they're pretty important to the food web. They're also ecosystem engineers. People often think of, you know, things like beavers manipulating their environment because it's the most active. It's the most noticeable. It's on a very much larger scale. But crayfish also manipulate their environment quite a bit. They'll move rocks around. They'll create spaces underneath rocks by moving rocks in and out. They'll move sediment around just to kind of make the habitat better for them. So on a micro scale, uh, crayfish are pretty good at engineering their environment as well. Within streams, they're very important to stream food webs. They can make up 50% of the invertebrate biomass in streams. And sport fishes consume possibly up to 65% of the annual production of crayfishes. So, they feed on about every life stage of crayfish, from the little juveniles that are a lot of times eaten by think bluegill, species like that, to the larger ones that are an important forage species for game fish such as largemouth bass, smallmouth bass, species like that. And they're this kind of key component in these food webs makes them what many people call a keystone species. They occupy multiple levels of the food web. They're decomposers, they're prey for mammals, things outside the stream, things within inside the stream. They also break down vegetation. There's a saying in the crayfish world that crayfish eat everything and everything eats crayfish, and it's pretty true. So kind of some of the general habitat needs. The one big basic need is water. If they can get to water, there's a chance that they can, some species can probably thrive there. Some of the stream dwellers, some of our SGCNs need a little bit higher quality habitat, like you see here on the left. Other species are okay with just these little roadside pools or these little intermittent streams where, they're seasonally have one pool for several meters. But one of the downsides is some of these habitats that used to look like maybe this on the left now look more like on the right. This is a photo from the Little Wabash River where the newest T&E crayfish occurs, and it's a lot of their original habitat is gone because it's just through channelization and siltation. So crayfish are one of the most threatened species. Freshwater mussels get a lot of attention and rightfully so. But crayfish are a close second. This was paper was from 2000. This number has actually gone up. Usually now when species are described, they typically have narrow ranges. So this number has probably gone up a decent amount. But from this graph, you can see they're one of the most threatened taxa in North America. I mentioned some of the threats, such as pollution, either from sedimentation, agriculture practices, mining. Basically, it releases a pollutant into the water or a chance of a pollutant into the water. It can have a negative effect on crayfish. Habitat alteration and water extraction, groundwater pumping in some states has caused base flows and streams to completely dry, completely wiping out entire wetted sections of stream. There's no water, crayfish can't survive, as well as things like habitat alteration, such as damming still has effects on crayfish. Gravel mining was a big one historically from people moving river rock out of the streams, channelization, and sediment, increased sediment loads. And then the other big one that is kind of really more the focus for this project, invasive species. These guys are probably one of the biggest threats to native crayfish because of how frequently, historically and currently, they have been moved around. So, Illinois is not unique in having invasive species, especially invasive crayfish. Most states have some sort of invasive crayfish species. In Illinois, the two that we're concerned with is Orconectes rusticus or Faxonius rusticus now. This species is native to, I believe, the upper Ohio and has been spread by bait bucket introductions to much of the Midwest and West. The other species in Illinois is Procambarus clarki. It is native to, as I mentioned earlier, it's native to extreme southern Illinois, but it has been introduced in the Chicago area in a few isolated locations outside of there, and it seems to be spreading. So in Illinois, invasive species, rusticus was first documented in 1973 from kind of here in north central Illinois. And since then, it has spread. Recently, we did some surveys and we found it moving within the central Illinois River, as well as Salt Creek here, not too far from Champaign. Uh, Clark Eye was first documented in 2001, though there are some reports that it may have been around a little earlier. The first one that we have in our collection, the first documented collection that we can verify, was 2001. Past few years, we've documented populations down here in southern Illinois at Rend Lake, which is just outside of its native range. And then we've picked up some that we're using as bait, that were being used as bait um, at Clinton Lake, and that's one that may become an invasive. We haven't documented established populations yet, but we are definitely keeping an eye out. So from 1973 to 1984 um, was kind of around the initial findings of rusty crayfish when it first started popping up. Again, it was kind of mostly centered around, those early collections were mostly centered around kind of northeast Illinois, kind of the Chicago area. But since 1984, it has spread to most of northern Illinois. Most drainages in northern Illinois, a lot of streams. The species is present. Some of these are new or bait bucket introductions since 1984 because not all these systems are connected. And then some are just normal uh, dispersal from the species. So, getting into some of the sampling methods that are used for crayfish, it's really pretty basic. One of the methods that we use here most is kick seining or drag seining, similar to what you would do for fish. This means setting up the seine around some sort of habitat, like here in the upper right picture. This is a seine set around some aquatic vegetation. This is not Illinois, this is Texas, but still, same premise around aquatic vegetation. And then you just disturb that habitat, create some flow if there is none, that pushes it into a bag or a little kind of a bag that you've created in the seine. This is the method that we use most often. The downside is it requires multiple people. It's also a good method for if you have flowing water because you can set downstream of habitat and disturb the habitat to push the species downstream, which they'll naturally go to when you disturb. Another method is dip net, similar to um, other like the standard D-frame dip net that's used for other macroinvertebrate sampling. There's a couple different methods that have been used for this. You can do the standard kind of um, standard macroinvertebrate sampling where you just kind of dip in or jab. And the other method is you can again set upstream below a substrate area or some vegetation and just kind of a smaller area, kind of gently swoosh um, water into the net, and crayfish will hopefully be in there. This method is you get a lot more juveniles with this method. You get mostly juveniles with this method, but you can occasionally get some adults. The other option is hand collection, where the water's clear enough or shallow enough. You can really find some um, um, can really find a lot of crayfish that way. And that's, I'd say, a lot of what the iNaturalist, current iNaturalist ones are from just hand collection, flipping rocks and picking up crayfish. Sometimes if you go out in the evening or at night, the headlamp, they'll just be out wandering around as well. So some basic taxonomy of crayfish. I'm not going to go into detail for all these parts. It's way too much information. Um, but some of the information that's good to know that helps with identification are the chelae, which are what we all call the claws. These are technically the first walking leg, so the first of those 5 paired legs. Um, these are the chelae, claws, whatever you want to call them. Another good part to remember is the rostrum. So kind of this part of the exoskeleton here towards the head. And then the carapace, which is this midsection here. Um, but mainly with the carapace, the area that uh, is kind of important for identification and narrowing down species is this area between these two areas of cervical groove here in the middle. This area is called, I think it goes actually. Yeah. This area here is called the areola. And the width of that, whether it's completely pinched off or if it's wide, that area will also help identify species or help narrow down what species you may be looking at. The other key characteristic for males and females is the reproductive parts here on the underneath side. In males, there'll be these two protruding elements, and females, you have a single annual ventralis. And here's a better picture of those. So on the photo on the bottom left, that is a male. The photo on the bottom right, it's hard to see, but that is a female. You can see the two gonopods here within the male. And here's kind of a closed up version. Again, this right here is the annulus ventralis, the female, or the sperm receptacle. And then on the male, you're not looking at this part. This is just this is a modification to kind of help protect the gonopods. These are just a modified swimmerette, which is your swimmerettes down here. But then the male gonopods are right underneath here. And male gonopods, especially form ones, are key characteristics for identification. A lot of taxonomic keys are based on Form 1 males, which unfortunately you don't always get. So I've mentioned Form 1 and Form 2 males. So Form 1 is what's considered the reproductively active stage. So this is when you'll see this form here, and then over here, the other Form 1, you'll see these when they're reproductively active or they're out seeking females. And they'll molt in between these two stages at least once a year. Form 2, Form 1's the reproductively active. Form 2 is the reproductively inactive. And you can see that here on the left and with this one here on the third photo from the left. These are two of our species. This is Propinquus, which is a native species in the north that is largely affected by rusticus. And on the right is the invasive Procambarus clarki. The big difference between Form 1 and Form 2, it's easier to see here on the Propinquus on the left. Form 2, when it's not reproductively active, the best way to describe it is it just kind of lacks definition. You don't have the level of detail that you do with the Form 1. You think about it, they have to deposit sperm fairly precisely, so they need some flexibility. So these Form 2s are very rigid. They're not very flexible, where Form 1s, each of these processes has some flexibility. With Procambarus species, it's a little more difficult. I'll admit, there's times that I still struggle sometimes to notice the difference between Form 1 and Form 2. Luckily, with clarki, it's not really necessary to be able to tell if it's Form 1 or Form 2 to get it down to species. But from here, you can kind of see you have this kind of amber color, similar to what you have over here on Propinquus. Kind of have this amber color here to the tips, and then kind of down here in the central, and then you'll have this long process will become a lot more noticeable when it's in Form 1. It'll be almost translucent. So we'll go over some of the genus differences. This will again help you narrow it down to a specific species. So again, as I mentioned earlier, all these photos I took in the last couple days with my phone here in the lab, just to show that you can still get pretty good detail um, using a phone or whatever you have with you in the field. I'll admit it's a little easier because these were historical, these were preserved, so they didn't necessarily move a whole lot or at all. Um, but so here from left to right, we have the native Faxonius propinquus. You see the two really short processes, and I should mention these are all Form 1s. So that reproductively active stage, you have propinquus here with the two short processes. This is another native, Faxonius immunis. These strong curves within these gonopods. The third is Faxonius virilis, another native species. See much longer, but also has some slight curvature there towards the tip. And that secondary process, the one on the bottom, is quite a bit shorter than the primary process, where that wasn't the case with the first two. And then the last one here is the invasive Faxonius species. This is Faxonius rusticus. Um, the species' gonopods are most oftenly confused with uh, virilis. They look pretty similar. Uh, the one, the main big difference here is you will not see uh, virilis's gonopods, if you get a large male, are typically much larger when compared to a specimen of the same size of rusticus. And on rusticus, these uh, there's not a whole lot of curve within this primary process or even the secondary process. They stay relatively straight. In the secondary process, the one down here on the bottom, it almost even kind of curves slightly back up towards the primary process. Again, so going into a little more detail. So again, I mentioned propinquus and rusticus. Rusticus is kind of displacing propinquus across a lot of propinquus's range in northern Illinois. The big difference here, other than the Form 1 and Form 2, so even on Form 2 males, you can still tell a pretty good difference. It does look a little more similar in Form 2, but propinquus here on the left, even at Form 2, you have two very short elements, and they are still relatively the same length, where with the rusticus here on the right, even the Form 2 male, the primary process is still a little bit longer than the secondary, and they're even at Form 2, they are both longer than what propinquus's is. If you have a female and you don't want to, or you don't want to try to uh, look at the gonopods of the male, one of the characteristics that you can look is looking at this rostrum that I mentioned earlier. With propinquus, there will be a structure called a carina. It is this little ridge that runs down the center. You can see it here in both these photos. It is a little ridge that runs down the center of the rostrum. And you can feel this by taking your pencil or if you have a little bit of a fingernail, you can run your fingernail through it, or just anything that you can kind of have some sensitivity to within that rostrum area. And you can feel that ridge pretty regularly, especially on larger individuals. And here on the right, you can see this is a rusticus individual. You don't see that carina there in the middle. So that's kind of a quick way um, for these two species. The other species that I mentioned is often confused with rusticus is virilis. These two look pretty similar. On smaller individuals, you can see how their gonopods can be confused. Virilis with gonopods are going to be Form 2s and Form 1s. You're going to have more curvature within these gonopods for virilis, Form 1 and Form 2, where rusticus are going to be more straight. And then with rusticus with the chelae, the dactyl or the claw fingers essentially are going to be much longer when compared to the palm, this kind of section here from this movable finger to the base. The chelae on larger individuals is going to be, that proportion is going to be much higher when compared to virilis that is quite a bit shorter. The chelae are more broad. Um, Also, one of the other characteristics is not just these photos, you do see it um, with the tubercle patterns. Virilis often will have a little bit more, um, a few more tubercles, more abundant tubercles along these inner margins of the chelae. But when all else fails, you can always look for that rust spot. Uh, that holds up most of the time. I will say when things within some of these systems, um, I think somebody in chat mentioned they can hybridize. Propinquus and rusticus have been shown to hybridize. I'm unsure how common it is, but it has been documented occurring. Um, uh, Rusticus has been shown to hybridize with Propinquus, and I also believe Virilis as well. Um, so it's it happens. I don't know how common. It hasn't been looked at a lot, but if we have noticed it, it probably happens more than we think. So in that case, sometimes if it is a hybrid, you may not get that rust spot, um, but you'd still hope that you get some of those other characteristics. The other species that I didn't mention that's often not confused with rusticus is Vaccinius immunis. Again, these have these strong, even in Form 1, Form 2, these strong curves on the gonopods. They'll also have a notch in the chelae that's pretty noticeable. You can kind of see it right here. And another thing, when these things are in their normal colors, they almost have an hourglass shape on the carapace that's different colors. It's pretty noticeable. So Procambarus gonopods. So on here on the right is the invasive. That's Procambarus clarki. In the center is one of the natives, Procambarus acutus, or the white river crayfish. And then on the left is the Procambarus burrowing species, which is Chrysalis, which is one that's often hand collected after rains. And the big difference between these three, to try to tell these apart, is the prairie crayfish or Chrysalis. Uh, their gonopods are mostly straight. Acutus, there's going to be some curvature to the gonopods. You'll notice that in Form 1 and Form 2. Same with Chrysalis. And then um, Clarki, the only way that I've ever been able to describe this is it's almost kind of like a, a loosely closed fist with your thumb sticking out. You think of these as kind of as knuckles. It's almost like a fist. It's just a kind of a ball of different processes that don't really have that strong curvature that you see with acutus. Outside of gonopods, there are other ways to tell Clarki and acutus apart. One of the best ways is looking at this areola width that I mentioned earlier here on the carapace within Clarki. It's going to be very, very narrow, almost completely, almost non-existent. These two um, parts of the carapace will almost touch. Underneath here are the gills. Um, Same with the cutis. You have a little bit wider. You can see there's a noticeable margin there. Admittedly, that's a little more difficult in smaller individuals, but when they get larger, it's a lot easier. Another characteristic is the tubercles on the chelae, especially on large males. The tubercle patterns on the chelae will be a lot more pronounced. Tubercles will be a lot larger, a lot more um, coverage on the chelae pads themselves than they will on acutus. Um, some of them I mentioned the gonopods, so you can kind of see here the chelae as well again, and then that gonopod structure even in Form 2, kind of see that kind of almost just kind of compact ball fist-like structure here on Clarki. The cutis, even in Form 2, and this is a smaller individual, you can kind of see that curvature. Cutis males will often have these little um, hair-like structures kind of around them as well. That's usually pretty noticeable. Then gracilis, not always confused with clark eye, but it can be, especially when you catch one of these guys out wandering around instead of in a burrow. The big difference here is that straight gonopod, not a lot of curvature. Pretty much everything on that just goes straight out. Another big tell is the chelae are a lot different shape. So again, like other burrowers, they have this thin outer margin that's used for moving, pushing dirt, keeping their burrow clean, and then the inner tubercles will be much larger and white on the inner margin. And over here, you can see on this individual, they're wider. And on the carapace, that um, areola is almost non-existent, even more narrow than it would be in uh, acutus. Then the last two are Lacuna cambaris and Creaserinus. These are the other primary burrowers. These are exclusively burrowers. Um, both these genuses are made up of exclusive burrowers. So Lacuna cambaris is the one that's been recently split. Here you have what used to be Diogenes, but is now Nebraskensis. Form 1 male. Here's the polychromatus. Uh, for the male gonopods, there's just a little bit more curvature, a little bit larger gap between the two processes than in polychromatus than in uh, Nebraskensis. And then in Creaserinus, it looks a lot different. You have pretty much no gap in between the two processes, and the secondary process kind of comes out at a weird, almost a right angle, and is much longer than the primary process. Looking at Polychromatus and Nebraskensis from above, um, these two species can be very difficult to tell apart. Um, the first things you can do is look at body shape. Polychromatus is going to be more com laterally compressed. They're going to look more, my boss always kind of look more bullet shaped. They're kind of more round, where Nebraskensis is going to be broader. It's just going to be a much bulkier looking crayfish. And then you can look at the chelae. Um, Nebraskensis will typically have longer fingers compared to the length of that palm. If you can see here, the one on the right, this finger compared to this palm, this finger is much longer than this palm, where these chelae for the polychromatus are much more compressed or kind of squat. And then the other one, it's hard to see, is this tubercle pattern within polychromatus. You have uh, you have a kind of a row here, and then they're kind of intermixed within here without a whole lot of open space. With Nebraskensis, you have a row here, then you have a small row here, and often right down in this area towards the base, you'll have an empty spot without tubercles that you don't normally see with polychromatus. The polychromatus will typically have tubercles there. And then Phodians, again, that gonopod's quite a bit different. This species also has a big notch on that dactyl that's pretty noticeable across its range. And this is one of the only species in Illinois that lacks a optical spine here. Most species have a spine that comes off right here, and Phodians does not. So if they lack that, it's a pretty good indication that it's Phodians. These spines are also good just to tell between a burrower and a stream dweller. Burrowers, you think they have to be moving in and out of these burrows constantly. It's best not to have things that will catch. So burrowers often lack this cervical spine that's right here that's on a lot of crayfish. Some crayfish even have two or three. So most burrowers will lack that spine. At least I can't say all, but the ones that I have encountered do lack that spine, at least the ones in Illinois. Either will lack that spine or it'll be greatly reduced to where it's more just a nub. And they also lack spines on their rostrum. So they kind of have this more shovel spade shape compared to um, Clarkeye and Fearless, which have these kind of spines on the end that kind of offset what is called this tip, which is called the acumen of the rostrum. One thing to kind of look out for, because I'll mention taking photos of chelae, is regenerated chelae. How you tell these is usually if they have one original and one regenerated, the regenerated is going to be much smaller. If they're both regenerated, most species will have some sort of tubercle patterns within here, within the inner margins of the chelae or the pinchers. Um, when it's a regenerated, it's much more homogeneous. It's usually a straight line of chelae on the top. I mean, straight line of tubercles on the top and a straight line of tubercles on the bottom. There's not a lot of um, heterogeneity with the tubercle patterns. It's pretty uniform. All right, so now we'll kind of get into the photos real quick. Um, these are pretty straightforward, so I'll kind of get through these pretty quick. One of the first ones to take that's really important is this dorsal view. So this is just a top down. This really helps us look at color patterns, and when these are taken well, just with normal magnification, we can see the areola here, we can see the rostrum, see the areola, we see the areola here, the rostrum. We can tell if there's spines. We can get a lot of information just from these good dorsal views. The big thing here is the color pattern. It's good if you can include the abdomen. It's not always necessary, but if you can, try to include that because there is some pattern and banding that can help with identification on the abdomen. The other is this lateral view. So this will kind of let us see the difference in the color patterns along the edge. With rusticus, this will allow us to see that rust patch. um, Also, this will let us kind of know of body shape. Some species are more compressed than others, so they're flatter. Rusticus, for instance, is usually more compressed or dorsally compressed than Virulus. So that's another way you can kind of tell them apart. Rusticus tends to be pretty flat on top, where Virulus still has some it's still pretty round up top, where Rusticus is pretty flat. So these lateral views, that's something we can look at there. We can look at the spines here as well. Chelae, there's a couple different ways to do the chelae photos, like the one here on the left. You can isolate the chelae and take a single picture of one chelae. Try to take a picture of the original chelae if it has them. They don't always have chelae. I get it, that's fine. Sometimes when you hold them a certain way, you can get them to just flare out both chelae. This is perfectly acceptable. These are two regenerated chelae actually, because you can see that there's no inside margins are pretty homogeneous. And then when they're juveniles, you can almost just kind of pull them around because these aren't really going to hurt you. But again, as some of those things I mentioned earlier, chelae morphology, shape, also tells us a lot about what species it could be. Then finally, the rostrum. This is a good photo. Most of the time you can get these photos just by um, most of the time you can get these photos just by taking your dorsal photo, and as long as it's of good quality, this is the same photo just zoomed in. That's perfectly good. I edited this right on my phone, cropped it to that level, and then just uploaded it to the computer. Same, this is the previous photo. Here we can see the rostrum and the chelae shape. That's perfectly acceptable too. So this one you don't always have to take another picture for. If your dorsal picture is good and the chelae are kind of out, you can use that photo as well, and we can work with that. It works perfectly fine. And the final is the gonopods. So females are pretty straightforward, just a straight photo of that annulus ventralis. Males, if you're comfortable with it, it helps if you can pull out the gonopods, whether it's form 1 or form 2, doesn't really matter. We can pull that out. Can pull those out a couple different ways. When you're in the field, if you have tweezers, or forceps. Forceps work great. A lot of us don't take forceps in the field. So another way that I found that works really well, if you're using a mechanical pencil, you can break off a little piece of pencil lead and kind of um, poke down in there and pry one and pull one up. Doesn't hurt the crayfish. And then you can leave that piece of pencil lead kind of in there, and it kind of will. I'll show an example here in a little minute with a preserved crayfish, but you can put that piece of pencil lead kind of in between down here at the base, and it'll kind of hold that up so you can photograph. That works for most species. If you're not comfortable doing that, you can't really get that gonopod picture out. A Just a picture of the gonopods while they're still um, up next to the individual will work fine if it's done from this similar kind of um, top-bottom view, kind of like what you would do with the female. So I saw there was a question about handling live crayfish, so let me stop sharing my screen here. So I have a crayfish here. It comes down a little bit here. So this is um, Babe. She's a blue gracilis that was given to us by a person in um, a person in St. Louis. So she's a little unique because she's blue. So some species will change, have different color morphs. St. Louis, for whatever reason, has these blue color morphs. So one of the best ways to pick crayfish up until she's a little, let me try to turn off my blur here. Okay. So one of the best ways to pick them up is you just want to go in here behind the chelae. The chelae are the only thing really on there that can affect you. So if you go in and just kind of grab them right here behind the head, you can kind of hold them up here, keep your fingers back. She can't get to me now. So when you're taking photos, once you have them like this, you can kind of manipulate them with multiple hands. So if we take a dorsal view, you can kind of reach up here, and if you kind of slightly push down on the bottom edges of the arms of the chelae, and then you kind of can hold, oh, what's freaking out? You can kind of hold the body like this, grab my phone, and then with that, take your photo, you have your crayfish there, and you can snap your photo of the dorsal view or of the side view. And then of the top view, same thing, kind of hold it like that. Keep your fingers out of the way. You have a good top view. Then when you have to do the chelae and the rostrum, you take her with another hand and kind of do this. And a lot of times when you do that, they will throw their chelae up like that. A lot of species will. A lot of specimens will. When they do that, you kind of just hold those chelae in place like so. She still can't get me. And then you just use your phone again, and you have a good uh, top view that shows the chelae perfectly well, and she still can't get to me. One of the best things I can tell you about this is just pick up and handle crayfish. You know, start with a small one and work your way up to bigger ones until you're comfortable. I would not start with a burrowing species because they can be a little more, they have a little more grip strength than stream dwellers, and they can't hurt. Um, so when it comes to doing males, let me grab a specimen here real quick. Comes to doing males, this is a preserved Clark eye. Comes to doing males, you'll have these gonopods here, right here with the males. You can kind of get underneath there, and I can, um, after this, I can pull up the microscope and share my screen there and show you that. But what you can do here is you kind of pull up the gonopods right here, can pull them up with a piece of lead or a stick or anything. And then you can kind of see that right there, that piece right there. And if you leave the pencil lead in there, it'll keep that gonopod up like so. And you can snap a photo It may take a couple times with obviously with preserved specimens, it's easy because they're not fighting back. But with this is a method that I use on live specimens in the field as well, and it typically works pretty well. It may take a couple tries to get exactly right, but it works. Another option is if you have somebody in the field with you, you're not by yourself, you can just pull that one up and have somebody else snap the photo while you hold that process up. Or that gonopod up works just as well. Um, so with that's kind of the photographing thing. I can, if anybody has any other questions or wants to see anything specific of any of the taxonomy or things like that, I do have the microscope here, and I can uh, share my screen to show that if somebody wants to see that. Um, if not, I will take any other questions or I'll take questions and also do that.
Can I
So
cover my
whichever.
slide real quick?
Yeah, go for it.
Okay. My computer has restarted, so I don't want to jinx it, but I haven't been kicked off since then. So I'm just going to start with the iNaturalist slide. So we talked about you'll have to make an account for iNaturalist. It's pretty simple, just setting up an email with them. And then to find our project on the website, it's under this community tab. And then there's a drop down and it says projects, or if you're on the app, it's on the left side of the screen. There's like a menu you can open that says projects. And if you search Illinois crayfish, there's two different projects that'll come up. So ours is this top one that says Illinois Crayfish Finders, and it's got the RiverWatch logo on it. So this is our project page. This is where the recording will be posted afterwards. And if Dusty wants to share slides, we can put a link for that there too. And so you can see on our project, we've already had 51 observations. You do not have to join our project to participate in iNaturalist. So basically, as long as you submit an observation of a crayfish in Illinois in 2025 or 2026, it automatically gets added to our project. So you don't have to do anything special. You're just using iNaturalist to take these crayfish pictures, and then it's added to our project automatically. So these are the project requirements, and you'll see this on the project page as well. Again, it's just a picture of a crayfish is added in the time frame in Illinois. And so what we're hoping will happen after this training, after you guys have just learned how to take those pictures for identification, we're hoping that this percentage of what is research grade observations will go up. Because even if you aren't confident in identification, we'll have people looking at them, specifically Dusty, to help do the ID on all these pictures. So as long as we're getting good pictures of the crayfish, you don't necessarily have to do the identification while you're out in the field. And then this is what the observation page looks like on the app on your phone. So I don't want to spend too much time on it because I know a lot of people who volunteer with RiverWatch are already using iNaturalist. But it'll ask you to add your media first, and you can add more than one photo here. So if you have multiple photos to add, that's where you'll do it. And it works the same way if you're using a computer instead of your phone. It'll give you suggestions for an identification. Sometimes those are helpful and sometimes they're not. But when in doubt, you can just put it in that family Camberidae, and it'll just go to our project and we can figure out the identification later. And then it'll automatically select the date and the location based on the metadata on your photos, but you can manually change those if you need to. And then that's basically all you need. You do not need to add it to a project. Again, it'll just automatically get added to ours. So you just submit an observation like normal, and then you're good to go. Okay, that's all my slides.
Yeah, as Hannah was saying, uh, yeah, those photos are very helpful. That's one of the hardest things with crayfish that come up on iNaturalist is sometimes, we'll just get a photo of a chele or a photo of just a dorsal view, and there may be two or three species that look very similar. Unless we have kind of a multiple pictures, multiple angles to go off of, um, it's very hard, um, while being very subjective, to get to a 100% confident in ID. Luckily, in Illinois, it's a little bit easier compared to states like Alabama or Georgia or states in the Southeast that have 70, 80, 90, 100 species. So, but it is either way, it is helpful. And I would encourage you to um, try to identify it. It that's one of the we're all here and in this stuff to uh, because we enjoy being outdoors, we enjoy this science, we enjoy the ecology. It's a fun challenge. Like I said, I don't do a whole lot with plants or amphibians, but I'll go out, take pictures, and go back and pull out all my guidebooks and still try to ID it. It's a lot of fun. So there's a couple questions. Um, so the best time of year for crayfish activity is generally in the spring to early summer, or throughout the summer, and water temperature can have an effect. So in the winter, crayfish, especially in the northern half of the state, will be pretty, they don't really hibernate, but they become very inactive. You can pick them up and even when you pick them up, they just don't move. Um, and they're a lot harder to collect in the winter as well because a lot of times they'll get down really deep into substrate or deep into riprap or under a bunch of detritus or in coarse woody debris, anything that kind of keeps them within a lot of cover so they can stay there for a long period of time. Once that water starts warming in the spring, we typically try to go out um, mid-May, late May. If you're in southern Illinois, that's a good time to go out. As you work north, it's going to be pushed back, probably up in northern Illinois, probably start uh, going out early June to mid-June. Um, but year-round, really, you can collect crayfish year-round, but that early spring, early summer, as long as flows are not too high, don't don't go out if flows are high. It's not safe. And plus, it's just not when flows are too high, it's just really not effective. It's not a very effective time to sample for any species, really. So, is there not too technical dichotomous key that can be used? Um, there are some online resources within Illinois. There's a couple publications. I think this one may be still available through um, PRI for the Illinois Natural History Survey. I think you can still get this one. This one is just a general field guide for Midwest crayfish, so it's not just for Illinois. You can see kind of here with the different chelae, different morphometrics. And these also have range maps. This is a great book for just general survey stuff, and I don't think they're too terribly expensive. But these also have a beginning. These will also have find it, also have your typical kind of dichotomous leaflet key to do that. More readily available, and I believe free, are these. These have the invasive crayfish. These are put out by um, the Michigan State, and I think partially with Great Lakes Restoration Initiative and some of those, and the EPA. These will help you ID the invasive crayfish. So this kind of folds out and kind of has some kind of general and folds out to a poster. You have Clark eye and the kind of the characteristics. Uh, rusticus here on the left, And then the bottom two are species that have not been documented but are on uh, the watch list for a lot of aquatic species. So that's that one if you just want to be able to kind of narrow it down between invasive or not. Oh, yeah, I can show that again. Uh, so yeah, this was put out, I think this is 2014, so it's a little it's getting a little dated, but the information's still good. It'll still get you to where you need to be. Other than that, there are online resources. Um, they're not as common for fish, but I can tell you if you put the photos on iNaturalist, I will look at them, and there's a decent following of crayfish people on iNaturalist that will also put their uh, 2 cents in as well, which is always great. I like having people do these things. So um, but yeah, some there's also some of these invasive guides that are put out by I think the EPA and some state agencies just have some basic key characteristics for these species. And we will make this presentation, I believe, available on the iNaturalist page, so you can go back and review that. Um, and yeah, if you're in Muhammad and you need something ID'd, um, shoot me an email to make sure I'm here, but I'd be uh, happy to take a look at it.
I might have missed it. You might have answered this, but Rick also had a question about releasing the crayfish back afterwards.
Yeah, so if it's if it's an invasive species and you're confident it is a rusticus or a clarkii up in the Northeast, um, yeah, you probably shouldn't release it back. You need to probably humanely um, dispatch of it and do that by, there's different ways to do it. You can take a probe or something and separate the head from the nodal cord. Um, or, you know, I don't, I work in the museum, so in the crustacean collection, so we doctor some specimens. So I don't like necessarily killing things when it's not for science and I don't necessarily have to. Um, so keeping returning one individual or killing one individual, unfortunately, ain't isn't going to make a big difference if you're not comfortable with dispatching of the species or dispatching of the specimen. I totally understand that. I don't want to make anybody do something they're not comfortable with doing. Um, the one thing I do ask is just if it is uh, any species, native or not, please don't move it from the stream or from where you collected it. Um, it can be very, as we found out with a lot of the invasive species, sometimes that's all it takes is just a few individuals to be spread. So yeah, if you're not comfortable with dispatching one, I completely get it. Um, but yeah,
Are either of those invasive species good for cooking?
Clarkeye. Yeah, so Clarkeye is that's probably one of the ways that it was introduced into the Chicago area was through consumers. Uh, Crayfish, these are the ones that are popular down in Louisiana. I think right now, well, that's coming up, I believe at the end of the year. That's when harvest season is in Louisiana's in this late in the summer. Yeah, so Clark Eye is very popular in the food industry. That's the one that they eat in Louisiana. That's the main one that's eaten around the world. That same reason is how it got introduced over into Europe. This is strictly a North American species, but is now worldwide. It is the most widespread invasive crayfish species. found now on almost every continent. It's really only originally native to the southern Mississippi River drainage. So, but yeah, if you catch them in high enough density, go ahead. Um, uh, again, I'd probably uh, dispatch of them before, because you're not allowed, I believe Clark Eye, you're not allowed to move live. Um, I'm not sure if you're allowed to move any of them, any species live now. I'm not 100% sure on that. I'm gonna know Clark Eye you're not, so you definitely want to, um, if you get a bunch of them, I don't see why you can't eat them. Just throw them on, throw them in a cooler with a bunch of ice, get them really cold so they're not alive, and then cook them. But if you take them, don't release them again. Make sure they're dead. Rusticus, usually species within the Paxonius you don't want to eat because there's just not enough um, tail meat or abdomen meat to make it worthwhile. So rusticus was probably introduced through bait bucket, through fishermen. How they believe it was introduced and spread.
Okay, are there any more questions? Did anybody want to see? I know it's already 8:16, but did anybody want to see the microscope stuff?
Must stop recording, but if anybody wants me to pull up the microscope, has anything in particular they want to see with any of the species I discussed, I'd be happy to stick around a little bit longer to do that.